Okay, this is going to be a video of a lecture on the Calvin cycle. So the first thing that you always need to know when you're talking about a biological process is where are we? So we are in the chloroplast, but we're in the liquidy filling of the chloroplast, which is the stroma of the chloroplast. So we're outside the thylakoid, but still inside the chloroplast, separated by that double phospholipid bilayer. So why do we bother to make sugar if we've got ATP? You can't store ATP long term. So we need to form a very stable chemical energy storage molecule. So when your parents were in school, the Calvin cycle was referred to as the dark reactions, kind of in contrast to the light reactions. But it turns out that it doesn't happen at night. And so there are three reasons why the Calvin cycle really needs light. There's an enzyme that we're going to be talking about called Rubisco, which stands for ribulose bisphosphate carboxylase. So you can see why we call it Rubisco. It's a very long name. But Rubisco is light activated. Now, it's not really collecting light energy and converting it into chemical energy. It just only works if light is reflecting off of it. Also, if you remember, there are two products of the light reaction that are needed to power the Calvin cycle. And if it's nighttime and there's no light bouncing off the chlorophyll molecules, then you end up with no hydrogen ion concentration building up in the thylakoid, and you end up with no ATP and no NADPH that you need to power the reactions. And then also, if you remember from your leaf lab, you learned that most stomates are closed at night. So if the stomates are closed, carbon dioxide can't get into the plant cells and up to the palisade layer, so you're not going to be able to have enough carbon to produce sugar. Remember, our overall reaction is 3 carbon dioxide molecules plus 3 water molecules gives C3H6O3, which is PGAL, which stands for phosphoglyceraldehyde, plus 3 oxygen molecules. Now, one of the things that this diagram or this equation doesn't account for is the fact that you actually use 6 water but produce 3 water. But because you are using more than you're producing, nobody ever writes the water molecules being produced on the right side of the chemical reaction. And if you don't take that into account, you'd realize that there are twice as many carbons in a carbon dioxide as you actually need for your sugar molecule. This diagram that we're about to show you does not show you where the waters are produced. And in fact, we've been searching for a diagram that shows where the water's produced, and we know it's produced, but we can't find where it's produced. This diagram is a simplified version. It doesn't have all the details, and it focuses in on the carbon skeletons. So there are hydrogens and oxygens, and these molecules are more complex than we're showing you, but we're only tracing the pathway of the carbons in this diagram. So first, we're going to start with 3 molecules of ribulose bisphosphate. So we have a phosphate group on either end. That's why it is ribulose bisphosphate. The prefix bi means 2. So there's 2 phosphates. Some textbooks will call it RUDP, ribulose diphosphate. Now, RUBP is combined with carbon dioxide. So if you have 3 RUBPs, 3 carbon dioxides can be bonded, 1 to each. So we get a 5 carbon molecule plus a 5 carbon molecule plus a 1 carbon molecule makes a 6 carbon molecule. This intermediate is so short-lived, it breaks in half so quickly that we don't even name it here. Most diagrams at the 9th or 10th grade biology level don't include the name of that 6 carbon molecule. But 3 6 carbon molecules break in half into 6 3 carbon molecules of PGA, which stands for phosphoglyceric acid. Or phosphoglycerate. And the 3 here tells you not how many molecules you have, but that the phosphate group is on the 3rd carbon in the chain. But this number here tells you we have 6 of those 3 carbon molecules. Now in this next phase, we have 6 3 carbon PGAs converted into 6 3-carbon molecules of 1,3-bisphosphoglycerate. And if you notice, there's an extra phosphate that wasn't there before on the first carbon in the chain. And in order to add phosphate, it requires ATP, our old friend. So we also have some energy being added to make that conversion. Now, in the next step, we make these 6 3-carbon molecules of 1,3-bisphosphoglycerate into 6 molecules of PGAL. In order to do that, we're going to need hydrogen. And the hydrogen is added, but you can't see it in this diagram because this diagram is only showing you the carbons in the chain. Now, we might be excited because we have 6 of our product molecules. We have 6 PGAL. But the problem is, at this point, the plant has only taken in 3 new carbon atoms. So we do not get to keep all of those PGAL molecules. In fact, if we stopped here, we would run out of RUBP for the rest of this reaction to continue over and over again. Now, with PGAL, the plant can make all kinds of compounds. It can make sucrose, glucose, sucrose, starch, rubber, proteins, lipids, and amino acids. And of course, if you're going to make amino acids, you're going to need nitrogen from the soil, but the plant can take the nitrogen from the soil and add it to PGAL to make amino acids. So now, for every 3 carbon dioxide molecules that we are added, we get 1 PGAL molecule that the plant gets to keep. Now, the rest of this is showing you that there are several steps, but that we have 5 3-carbon molecules that are regenerated into 3 5-carbon molecules of our RUBP, so we get our original 3 5-carbon molecules back, so that this whole entire Calvin cycle can continue indefinitely. In order to make that conversion from our 5 PGAL molecules, we need several different enzymes, one at each step of this reaction, but we also need energy from ATP. Fortunately, remember that the Calvin cycle is taking place in the stroma right outside the thylakoids, so we have plenty of ATP, NADPH, and ATP. So if somebody's talking about an energy donor that talks about phosphate being added, it's pretty much always coming from ATP. And if they're talking about an energy donor that's donating hydrogen, that's coming from NADPH. So which steps of this do you need to know? You really need to know RUBP, Rubisco, carbon dioxide, PGA, PGAL, ATP, NADPH. We tend not to ask you to know the name of 1,3-bisphosphoglycerate. And that's the end.